All right, I'm pretty sure I'm recording now. Oh, and someone asked, just to double check, do we have lab this week? No lab. You're free. Unless, well, no. Just kidding. No lab. Okay. Share screen. Okay. So I will try to see the questions, but oof, it's hard when I have a tiny MacBook and trying to fit everything on my screen. So if there's a question you really want it to be answered and I'm ignoring it, use your voice. Be like, hey, I have a question. Because then I will definitely hear it and I will definitely answer it. Okay. So the first topic we're going to talk about.
我进了 Zoom meeting，知道我的名字是什么吗？
Is that a question? Is someone...
陈硕。好，猪头是分享完了，
Oh,
我还在想他这个 TA 怎么把我放进去的，居然没有把我赶出去。
oh, okay. I muted them. All right. Where was I? Okay. People keep coming in. All right. So we're going to do plant growth / elongation. And what you need to know for that is, number one, what are the things that need to be accomplished for elongation of cells to happen? And then how does the signaling of that work? So there's kind of three things that need to happen. You need to get water into cells. So when water moves into cells, that's going to help them expand. Two, you need the cell wall to extend. So we need to increase the extensibility of the cell wall. And then three, you need the pressure potential or the trigger pressure of plant cells to be greater than the yield threshold. So the yield threshold is just the pressure potential required to do elongation. And her example of that was if you blow up a balloon, when you first kind of blow it up, it might not expand the actual rubber part, and then you blow harder and then it all of a sudden expands. So that kind of represents the yield pressure. So you can have water going in and you can have the cell wall increasing its extensibility, but if you're below that yield threshold, you're not going to extend at all. So those are kind of the three things required for a plant cell to elongate. So how are these things accomplished? So how do we get water into cells? Well, water is going to want to move from a high water potential to an area of low water potential. So if you have two plant cells, let's say one has a water potential of 4 and the other one has a water potential of 2. The water is going to move way more into the 2 cell than it is into the 4. Why? Because it has a lower water potential. Water moves high to low. So what determines water potential? Water potential is equal to solute potential plus pressure potential. And they have fancy little signs on, like, probably on your exam sheet. But I'm just going to do these letters because it's easier for me. So what do these things mean? So solute potential basically represents the influence of solutes in solution on how water moves. And basically you can think as you increase the amount of solutes in solution, so imagine making water super salty or something, you're actually going to decrease the solute potential. And if you look at the equation, if you decrease the solute potential, that should also decrease the water potential. So as you make something super salty, you're also decreasing the water potential and making it more likely that water will move in that direction. So if we go back to our plant cells, we could say this one over here probably has a ton of solute, way more than this one. And that kind of follows that you would expect more water to go in the direction of more solute, and less water to go in the direction where there's less solute. And it's just kind of weird that more solutes equals a lower solute potential. But then if you think of how that affects the water potential, it kind of makes sense. And if you're bothered by that more, you can ask me about why it's a negative. The other thing that influences water potential is pressure potential. And that's just the turgor pressure of the cell. I see there's a hand raised. Yes. Question.
Hi. So my question is just on during lecture and in class, she kind of discussed how ions can influence the solute potential, like potassium and calcium and stuff.
Yeah.
So I don't know, I'm just confused about like
That comes
what
up.
on the exam, like, are we supposed to be able to use the Nernst equation to find the solute potential or something like that? It's confusing to me.
Yeah, no. You should definitely know that, yeah, if you increase, and in the class, what we talked about, and the one that's going to be relevant for the test is if you increase the amount of chloride or you increase the amount of potassium in the cell, you're increasing solute in the cell. And if you're increasing solute, you're decreasing solute potential, and you're making it more likely that water is going to come in. So you don't need to calculate the amount of solute using Nernst or anything like that for this. What you might have to calculate is like water potential. If I gave you solute potential equals 3 and pressure potential equals 4, you should be able to use the water potential equation to calculate water potential. And then if I tell you which of these two cells is going to have more water going in it, you should be able to say, okay, this one has a lower water potential, this one has a higher one, there's going to be more water going into the lower one. So that's kind of the calculations you would have to do with water potential. And then kind of on an unrelated note, you will also always probably see some context where you will have to calculate Nernst equation, ECG, not necessarily for water potential, but just out of context. You should still, for this exam, know how to do that too. So we can go over an example if y'all want. Was there another question?
I was for the solute potential in lecture. I remember the professor saying that it's always negative. Like, does that mean a negative number
Yeah.
that would be given? Okay.
Yeah, and it has to do with just the solute potential equation is negative IRCT, or it's ICRT, something like that. So there's a negative side in front of it. And I think C, I haven't calculated this in a long time, but I think C is concentration. So as the concentration of solute goes up, it becomes more negative or gets smaller or goes down, which is why you get that kind of weird relationship. Okay, so I was saying water potential is solute potential plus pressure potential, and that pressure potential is just the turgor pressure. And turgor pressure is how much pressure is being exerted on the cell wall by the water in the cell. And so the higher that pressure is, the higher the water potential. So you can imagine the more pressure that's being put out, the less water really wants to come in. And if you decrease that turgor pressure, that's actually going to cause water to want to come in more and decreases that water potential. Okay, so that's one thing we need. We need water to come into cells. So we're going to figure out what is the cell signaling pathway to get that to happen. And like we kind of already mentioned, the cell signaling is going to have to do with increasing solutes in the cell, therefore decreasing the water potential and making it more likely for water to come in. But we'll talk about that. The other thing we need is the cell wall to extend. So we get the cell wall to extend by kind of breaking up the cell wall. And it turns out that's accomplished by increasing the amount of hydrogen ions in the cell wall. So that's also going to decrease the pH of the cell wall. So the mechanism for that is if you increase the amount of hydrogen ions in the cell wall, those hydrogen ions themselves in that acidic environment is actually going to just break up bonds that are keeping the cell wall together. And then in addition to that, you also get proteins activated that act to kind of cut up the cell wall. And both of those together will cause the cell wall to expand. So we'll talk about the cell signaling pathway that does that. And then the last thing is just you need the turgor pressure to at least be high enough above the yield pressure to get the elongation to happen at all. Did I see a raised hand?
Yeah, so I was gonna ask, is that expansion protein itself gonna break like the bonds between the cell fibers, or like hydrogen bonds are gonna be break by the hydrogen ions just present both ways?
Both.
So do
Yeah.
we have to mention like both ways inside our like say flow chart or so?
I don't think you would need to say both. I think if you said either one of those, it would be good enough. But
So we, okay,
I
so
would
we can, yeah.
every time she's mentioned it, I feel like she's mentioned the expansion proteins. So I would throw that in there.
So probably what we can do is just increase hydrogen concentration outside in the cell wall, and then the next step is probably going to be like increased activity of expansion, and then like maybe decrease bonds between the cell wall fibers.
Yes. Yeah.
Okay, cool. Thank you.
So those proteins that break up the cell wall are expansins. Yes, another raised hand.
Hi Rachel. So I have a question regarding like the Nernst equation.
Yeah.
So from what I understood, we use 59 for plants and then 61.5 for animals.
Yes.
But there's a question on the unit packet from chapter 7. They use 61.5 to calculate the gradient.
For a plant.
It's
It's a mistake. The unit packet has lots of mistakes, as it turns out.
okay. So like for this whole, for all the plants, we only use 59, right? So
Yes.
okay. Because on the equation sheet, there's like also mentioned about temperature.
Yeah. I think the temperature is what causes the change in the number.
Okay, so that means plants, usually the plant temperature is 25, which is room temperature. Oh, I see. Okay.
Right. And then our bodies are a little bit hotter.
Yeah. So, and then that's that's why animal use 61.5.
Yeah.
Okay, sounds cool. Thank you.
You're welcome. All right. Any more questions before I continue? So here's 3 things we need to get elongation to happen. Now let's talk about the cell signaling pathways in order to accomplish that. And specifically, I'm going to say, let's do the scenario where we have, here's my plant stem. And inside my stem, there's like a bunch of plant cells. And there is light coming in from the left. And so what cells do we want to elongate? The right side cells, right? If these cells on the right side elongate, then the plant is going to move towards the light. So how is that accomplished? So step one would be light detected by PHOT, which is phototropin. It's a pigment. So on plant cells, there's these photoreceptors called phototropin, and they get activated by blue light specifically. When PHOT is activated by light, it will autophosphorylate. When phototropin autophosphorylates, it activates a signaling cascade, aka you don't have to know the individual steps of what happens, but you need to know that in the end, that causes PIN proteins to move on the opposite side of the light. Opposite to light. So PIN proteins are they are proteins in the membrane that eject auxin. So I'm drawing them moved from not just all around, now they're on the side opposite of the light. And that'll be true for all of the cells. So for that second row, now they've all moved their PIN proteins to the right. This third row, move them all to the right. Yeah, I see a hand raised.
Yeah, sorry, this is just a quick question. Does auxin bind to the hydrogen pump, or does it bind, or does it cause another interaction?
It itself doesn't bind to the hydrogen pump. It indirectly is gonna change things with the hydrogen pump, as we will see. Raise hand. Maya.
Yeah, I had a question. So I remember her talking about in lecture that the PHOT,
Mhm.
the phosphorylated PHOT acts as a protein kinase, and I don't know what that means. Can you like
Yeah.
clarify?
So whenever you hear the word kinase, it's an enzyme that phosphorylates things. It adds a phosphate group. Whenever you hear phosphatase, that's an enzyme that takes a phosphate off. So kinases phosphorylate, phosphatases dephosphorylate. So PHOT is a kinase because it phosphorylates itself. Hopefully that makes sense.
Okay. Thank you.
You're welcome. Was there another question?
Hi.
Yes.
I remember her talking about oxygen and negatively charged oxygen in the lecture, and like us just didn't know like what the difference between those two.
About negatively charged auxin?
Yeah, like she just I don't know interchangeably used those words, and I don't know if there's any difference between normal oxygen and negatively charged oxygen.
No, I think she might have mentioned it just saying that auxin was negatively charged in the context of like predicting how it can move or why it moves in a certain way, but it's not important that it's negatively charged.
Okay, thank you.
You're welcome. More questions? I see hands.
Oh yeah. Oh, sorry, Sydney can go first.
Okay, I was just gonna confirm. So like this with the float, that's just happening like at the same time, but like independently of like the oxen doing the sour stuff. Right?
Yeah. Yes. Yes. Yes.
Okay.
Yeah. So I'm gonna get to oxen here in a second. Next
Oh yeah,
question.
and then oh yeah.
Yes.
Yeah, so with the negatively charged auxin, wasn't it that it couldn't move through the membrane if it was negatively charged inside the cell and it needed like some kind of channel to get out of the cell where it becomes positively charged again because there's hydrogen? Am I remembering that right?
Yeah. Yeah. So I was gonna say it has something to do with movement of oxen and that makes sense. It can't just passively go through the membrane. It needs a transporter, either the oxalates or the pin proteins to get anywhere. Because if it
Okay,
could move through the cells, then all of this would not make any sense.
yeah. And oxalates was auxin and chloride inside of the cell, or is it auxin and auxin H plus?
Yeah, it's a symporter for oxen and hydrogen ions.
Okay, okay. Thank you.
You're welcome. Okay. Any more questions? Okay. So now pin proteins are all on the opposite of light. Why does that matter? Because pin proteins are the ones that push auxin out. So if auxin gets into these cells, they're going to push the auxin to be concentrated on the right side. So where does auxin come from? So auxin is made in the shoot tips, and then it travels down plants, and it gets into plant cells through little proteins on the top of plant cells called oxalates. And oxalates proteins are active symporters that get auxin in by using the energy of hydrogen ions moving down their concentration gradient. So yeah, we could say at the same time this is happening, we also have auxin entering via oxalates symporter. And then, sorry. Ooh. Okay. And then auxin will move where pin proteins say. So that's going to cause auxin concentrations to increase on the right side cells. So now what do we expect happen? The right side cells are going to elongate. And how does an increase in auxin in those cells cause that? So next, auxin is going to do signaling that should sound really familiar. It should sound a lot like JA signaling from plant herbivory responses. So auxin is going to enter nucleus, Auxin will bind its receptor in the nucleus. And then the auxin plus receptor is going to go to DNA, and it's going to kick off a repressor of growth genes. So kind of the same thing as JA, except not the repressor isn't JAS and the genes aren't defense genes. And so one of those growth genes is sour. So the next step will be, sorry, people keep asking to come in. Next step will be sour transcription increases because it's no longer repressed. And what does the sour protein do? Sour protein inhibits phosphatase. And what do phosphatases do? The opposite of kinases. Phosphatases dephosphorylate things, where they remove a phosphate group. And why is this relevant? Because the hydrogen pump on the membrane of plant cells only works if it's phosphorylated. So if phosphatase is around, it's going to dephosphorylate the pump, and it's going to decrease the pump's action. But if we inhibit phosphatase, then we have increase in hydrogen pump activity. So auxin has resulted in increase in hydrogen pump activity. So what's that going to do? Hydrogen is pumped out of the cell to the cell wall. So we already kind of talked about what one result of that is. So one result is if we increase the ions in the cell wall, it's going to lower the pH. It will activate expansins. And all of that is going to increase the extensibility of the cell wall. We're breaking up bonds, breaking up parts of the cell wall, making it more likely to expand. Other thing that's going to do is what's happening actually in the plant cell. And that's going to hyperpolarize the plant cell. And it hyperpolarizes it quite a bit from this hydrogen pumping out of the cell. So we're removing positive charge. You can even imagine in your head that maybe it's going to like negative 130 millivolts. So it's becoming really negative. Now, what does that do? Well, one thing that does is it activates hyperpolarization activated potassium channels. So there are channels that if you hyperpolarize the membrane, they open. Now we're used to potassium leaving the cell, but if we're at negative 130, we're actually way below the equilibrium potential for potassium. So in plant cells, The equilibrium potential for potassium is usually around like 118 millivolts. So because the membrane potential is way more negative, there's actually a push on potassium to go into the cells. Because if we're here and potassium wants to be here, it wants to make the membrane potential more positive to get it to 118. My throat. Too much talking. Okay. So what does that have to do with cell extensibility? If we move potassium ions into cell, we've increased the solute concentration. And if we'd increased the solute concentration, we've decreased the solute potential, decreased the water potential, and made it more likely that water is going to come in. So that's one effect. The other effect is that there is hydrogen chloride symporters that are going to move chloride into the cell. So if we're pumping a bunch of hydrogen ions into the cell wall, basically what we've done is, this is our plant cell, this is the plant cell wall. You made it super high concentration of hydrogen in the cell wall, super low inside. So there's actually a gradient of hydrogens to move into the cell. So this symporter is going to move hydrogen into the cell high to low, and chloride ions use the energy from that movement to move into the cell. And chloride ions going into the cell is also going to increase the amount of solute in the cell, decreasing the solute potential and decreasing the water potential and making it more likely that water goes in. So all of this signaling. Oh, make sure if you just came in, you mute yourself.
Yes, that makes so much sense.
The Zoom meeting is happening right now for review.
How
How many people are there? 53, 55.
Okay, I found. Okay. So all this signaling, what did we do? We added 2 solutes, potassium and chloride, and then we put a bunch of hydrogen in the cell wall and made it expand. So yay, we've got cell extension just as long as we're also above the yield threshold. And then everything should be fine, and we have extended cells on the right side away from light. All right, questions?
Yeah, so I thought water potential remains the same, and it's just pressure potential that changes. Like I thought decreasing the solute potential will result in more water uptake by the cell, and that just increases the pressure potentials. I thought they kind of cancel out, and water potential remains the same. Like it's just that you increase the pressure potential that eventually lead to elongation. Can you like clarify on that?
Yeah, so it kind of works in a cycle, right? So as you have water moving in and as the cell wall extends, then yeah, you can get the fact that the water potential actually doesn't change because the cell wall is extending at the same rate that water is coming in. So you can get no change in water potential, even if you're initially changing it with changes to the solute potential.
So is it more accurate to say increasing the water potential changes the increasing the water potential changes the cell wall extensibility, or the pressure potential of the cell that changes the cell extensibility?
I wouldn't say, I don't think you could say either. I don't think you could say it's all about the pressure or all about the water potential. I'd say all of these things have to happen. You have to get water going in to a lower water potential. You have to be increasing the extensibility of the cell wall. So yeah, it's kind of all happening at once. You can't just say it's one or the other, and you won't have to say that on the test. You won't be asked like what's specifically. You won't be asked one thing because they're all happening. Other questions?
Yeah, I had one. Can you hear me all right?
Yeah.
It was about like on the slides, there's like a equation for like elongation rate. It's like ER equals M and then in parentheses pressure potential minus yield threshold. What proteins affect like what parts of that equation? Like M is the slope, like what proteins would affect the slope, or what proteins would affect like the yield threshold?
Yeah. So that equation was elongation rate equals M pressure potential minus yield threshold. If I remember it correctly. And the slope of that line was cell wall extensibility. So proteins that actually affect the cell wall would be like expansins. The turgor pressure or the pressure potential is going to be influenced by how much water is going into the cell. Therefore, it's going to be affected by the water potential and just how much movement of water is going in. And then the yield threshold can also kind of be changed by how flexible the cell wall is. So again, expansin proteins might affect that. But I wouldn't worry too much about changes to this equation for the test.
Okay, I was just wondering because on like the unit packet for growth, question 7 has like this big table thing where it like asks you about like what proteins change mass balance and like flux. I thought like maybe this equation would be like an easier way of like remembering it versus like just like hard remembering proteins and stuff.
I'm gonna say no. I'm gonna say this equation is not gonna be super helpful. I'm gonna say knowing this flow diagram is gonna be helpful. So you should be able to kind of follow how you go from like light being detected by phototropin to auxin causing all of these changes to the cell to cause it to extend. You should know that full pathway. And then you should also know just kind of these basics of water potential that I went over at the beginning. Like, could you calculate it just solute potential plus pressure potential? And do you understand that a lower water potential is going to have more water going in? Just kind of that basic relationship.
Okay, thank you.
You're welcome. All right, next question.
My question is, can you help me? My question is based on how much do we need to learn about the phytochromes?
Phytochromes. You don't need to worry about it.
Okay, we don't need to understand what it's doing. Okay.
No, I wasn't in lecture on Friday, but I can confirm that you don't need to know it for the test. I'm not sure if she got to it on Friday, but I can confirm you don't need to know it for the test.
Okay, so we just, okay, thank you.
Next question.
Hi there, can you hear me all right?
Yeah.
Okay, I was just wondering if you could repeat the part about the I guess the H plus and CL minus importers, which way the CL is fluxing and which way H plus was fluxing again?
Yeah. So symporter, they're fluxing the same direction. And you can think if we have this pump making it really high in the cell wall, really low inside the cell, and it's going to move down its gradient, then hydrogen ions are going to move in. And so symporter, that also means chloride is going to move in, which makes sense because we're trying to get these two ions into the cell to increase the solute concentration.
All right, thank you.
You're welcome. Question.
Yeah, I was wondering about aquaporins because I know that they were mentioned in the lecture that they're like on the membrane, but do we need to know much about them in detail other than just that they're there and that they have to, the amount of them kind of impacts the rate of water in or out?
Yeah. So yeah, you should know the relationship is if you increased aquaporins, you're increasing the amount of holes that water can go through. So it increased the amount of water in, decreased resistance.
Okay, thank you.
You're welcome. Next question.
Yeah, so my question is just on, so the hydrogen pump that's on the, is it on the cell wall itself or is it?
Between the, well, it's on the cell membrane and it's pumping it
Okay.
into the cell wall.
Okay, so then by putting more ions into this between the cell wall and the cell membrane, they are, so you're increasing the turgor pressure because you're putting more chloride because it's following the symporter, and then the potassium is flowing out of the cell membrane into the between the wall and the membrane.
Potassium is flowing into the cell, which increases the solute inside the cells. We're making it, you could think, have more stuff inside. So water wants to go in.
Can you just draw a diagram showing the flow of both of all of them real quick so I can, sorry.
No. And so here's the cell wall. So we have a hydrogen pump pumping lots of hydrogen ions in. So we have a much higher concentration in the cell wall than we do in the cell. That's going to cause a really, really, really low membrane potential. Let's just give it a number because it makes things easier. Say it's 135 millivolts. So that's going to activate hyperpolarization activated potassium channels. That's going to cause potassium to come in. That it increases the solute concentration, decreasing solute potential. And then also the pump's activity has created high hydrogen out, low hydrogen in. So we have a symporter here that is moving hydrogen down its concentration gradient from high to low. And it's a symporter that's also transporting chloride into the cell. And so this chloride, this potassium increases solute concentration, decreases solute potential.
Okay, so my question is just, then the water will flow in and will push the cell membrane up against the wall and that'll extend it?
Well, we need water to come in to make the cell elongate. But we also need the cell wall itself to break up so that as water comes in, we're expanding. Because if the cell wall is really rigid, if we put a bunch of water in through just this mechanism, we wouldn't actually elongate the cell, we would just increase the trigger pressure.
Thank you.
Cool. Next question.
I just have two questions. So does a stem cell have both hyperpolarization gated ion channel and depolarization gated ion channel for both electric signaling and this whole aux thing? Or not aux,
What did you say
the
about the
cell
the depolarization gated? Because we have
for the electrical signaling, like JA.
oh, for JA signaling. Yeah. Yeah. So if we wanted this plant to have a defense response, yes, it would need the regular voltage gated potassium channels too.
Okay. And my next question is oh yeah, I think in the lecture she said something about the plant root being negatively phototrophic. And I don't know, like I think she said like the phot pin relationship is kind of like opposite of those of stem cells.
Yeah.
I just didn't understand like what that means.
It means that when in stems you have phototropin signaling, the pin proteins move opposite of the light. In the roots, it turns out it's the opposite. It moves towards the light. It moves the pin proteins on the same side as light.
Okay, thank you.
You're welcome. That's an interesting fact, but I would focus on the stem flow diagram and the stem steps for the test.
Could you please talk about UP715? Just how all these skates are doing and what they do to the whole, depolarization and stuff of the plants.
So what I'm talking about here, there isn't much depolarization going on. There's hyperpolarization going on. I don't know. What is the question?
Unit packet 7, question 15.
What's the question?
So it's basically 2 cells, and then it's this Venus flytraps, and then
not on the test. No Venus flytrap.
Oh, for real?
Yeah, we're not talking about that. Yeah, we didn't go
Yes,
over.
let's go. Thank you.
Next question.
Hi, can you pull up the unit packet 8? I have a question on number 4. I don't really understand the graphs.
Let's see. Unit packet. Oh, so this is photos. What question?
Number 4.
Oh, yeah.
The one.
I was
yeah, the one
actually
with the graphs.
I was gonna go over those graphs after growth elongation. So any more questions about growth elongation, then we'll move on to photosynthesis and specifically those graphs.
Okay, thank you.
You're welcome.
Yeah. Do we need to know like how like the growth pathway works for like gravity? So instead of like light, would it just be like
no,
we don't? I bet. Thank you.
Hello, so at the very bottom, could you label where the symplast apoplast cell wall is, please? Because I was a little confused on where the pumps are pumping into, because I know the inside is a symplast, and that the hydrogen pumps are going to the cell wall, but then I'm kind of like, where's the apoplast?
Apoplast is just
Okay.
Out the cell wall, extracellular area, and Symplast is inside.
Okay, so when like the textbook or the worksheets say that it's pumping H into the cell wall, they kind of mean the apoplast. It's
Yeah.
kind of synonymous.
Yeah.
Okay, so there's also like all the cross chain fibers and whatnot in the apoplast as well.
Yeah.
Okay, thank you.
You're welcome. Yes, questions about elongation growth? Okay, cool. Before we do photosynthesis, I have to go pee. So I'll be right back in like 2 minutes. All right, I'm back. Okay, so
Actually, I have one more question.
yeah.
So do we have to know how phloem and xylem have different water potential, like how they have different mechanisms of water potential? And are they analogous to apoplast and symplast, or is it just like different thing completely?
Different thing completely. Apoplast is like outside of the cell, extracellular cell wall space. Symplast is inside the cell. Xylem and phloem. For this test, the only time it's going to come up is, I mean, you should know that like the water for photosynthesis comes from the xylem, and you should know that JA hormones can be transported to cells far away via the phloem. That's the only relevance for this test. And then we'll talk more about xylem.
So we don't have to account for the fact that xylem is composed of dead cells and that can affect different order potential. No, okay. Thank you.
Oh, hang on, quickly follow up on that. So apoplast is like actually outside of cell membrane, but like we're still within the cell wall, right?
Yes,
Just
yeah.
making sure.
It's the
Okay, thank you.
extracellular cell wall space, yeah. Prisha?
Sorry, just really quick clarification. So when we say that the H plus is or when the H plus is leaving into the apoplast and it's hyperpolarizing, does that mean that the potassium that's entering is just slowly coming in, or like how does that work?
First, we get the hyperpolarization
Mm-hmm.
that activates the potassium channels to then let in
Oh, then let in more. Okay, and then for the hydrogen and then the chlorine one, what cause or like why does the chlorine enter? Is that just it enters? Is that all we need to know?
Because when the pump pumps out hydrogen,
Mhm.
it's creating a high hydrogen out, low hydrogen in. And
Mhm.
the importer is going to move the hydrogens high to low, and it takes chloride.
So all we need to know is it just takes chlorine. Do we need to know like why chlorine enters or like the significance of chlorine?
Just that it's adding solute in the cell. So it's adding solute concentration, lowering solute potential, making it more likely that water wants to come in.
Okay, sounds good. Thank you.
You're welcome. Kara.
I had a question about like the overall content of the exam because there's like while reviewing for this exam, like there's like 4 different topics.
Yeah.
So would you say like are they all evenly distributed and like brought up kind of like in like a fair ratio between each other for the questions, or is it like
Yes. Yeah.
okay.
That's why, yeah, I said there's these 4 topics. What I'm going through is like this is kind of the core of what you need to know from each of those 4.
Okay. And I did have another question. When it comes to these topics, like a lot of them kind of seem to overlap a little bit. Like I can kind of see some similarities. Do you think we should prepare in a way that it's like we would expect different topics to be included in like a similar question?
No.
Okay. That's good to know. All right. Thank you.
You're welcome. Yeah, as you're going through the test, you'll be able to like categorize them as, okay, this is a growth question, or this is a photosynthesis question.
Okay, cool. Awesome. Thank you.
You're welcome. Another question?
Hi, I have a question regarding 1433 protein.
Yes.
So I thought technically kinase activate the proton pump, but I don't know what.
There's some.
Is it the protein that phosphorylate the inactivation gate, or is it the kinase?
I don't know the details of the pump phosphorylation. Yes, those proteins are involved. But what you need to know is auxin inhibits or causes sour to inhibit the phosphatase so that the pump can be phosphorylated. You don't need to know the details of what protein is phosphorylating the pump.
I see. So what do we need to know about 14, like 1433 protein? Is there anything that we need to like know about it in detail?
No, you just need to know that the hydrogen pump needs to be phosphorylated, and there's proteins involved in making that happen.
Okay, I see. Thank you.
All right. Then we will move on to photosynthesis and specifically the irradiance curve. So the irradiance curve has CO2 uptake on the y-axis, and it has light intensity on the x-axis. It looks something like this. Let's see. I should be straight. Well, oops, no. Oh. Now it's stuck on eraser. All right. Let me try to reload the page. It's like shutting down. It's like, this is enough. This is enough physiology for one day. What is happening? All right. You're being so slow. Hope it's not my internet connection. Hopefully y'all can hear me okay. Yeah, okay. Back. Draw. Yay. Okay. This one's gonna be better. Oh, look, that was so smooth. Okay. So the irradiance curve, what does it relate? As you increase light intensity, the amount of CO2 uptake or the amount of CO2 consumption over those different light intensities. So there are different points you need to know and understand on the graph. So the first point would be this one right here. So that first point represents the amount of CO2 produced by cellular respiration in the dark. So light intensity 0, CO2 uptake is negative because it's just cellular respiration happening. The next point you should know is right here. And that is the compensation point. And the compensation point describes when the amount of CO2 produced by cellular respiration equals the amount of CO2 consumed by photosynthesis. And that's why CO2 uptake is 0. Okay. Third point you should know is kind of what's happening when our curve's slope is changing. So that whole section where the slope is changing, that represents the light dependent section. And you can think more and more chlorophyll are coming online. So in that region, we're increasing the amount of light reactions, increasing the amount of ATP and ADPH we're creating. And then the last point would be where it plateaus. The plateau kind of represents maximum amount of photosynthesis, and you should think of that region as being things to do with the Calvin cycle. So the point at which photosynthesis becomes maximal is controlled by things to do with Calvin cycle enzymes or the amount of CO2 around. All right, I'll stop for a second and answer questions. One of you guys want to say a question?
Hi, I had a question about the compensation point, which is on the y-axis. I was wondering why when we increase cellular respiration, the compensation point lowers rather than goes up.
And by lowers, which way do you mean?
Sorry, not the compensation point. I meant the point number 1. Yeah.
Oh, okay.
Yeah.
Yeah. So that kind of leads into what I wanted to talk about next. So that's good. So yeah, this point number 1, if you can think of like above the x-axis as being positive CO2 uptake, so we're actively uptaking CO2 and decreasing the amount of CO2, then below the x-axis, we're doing the opposite. We're actually producing CO2. So that amount there actually represents the amount of CO2 being produced by cellular respiration. So if we increase cellular respiration, we would increase the amount of CO2 being produced, and that point would move down to represent that. That makes sense?
Okay, I think that makes sense. So you're saying that the CO2 point goes lower when you increase cellular respiration because you're increasing the amount of CO2 produced?
Yes. Yes.
Okay, thank you.
Yeah. So it's kind of helpful to kind of imagine some numbers on this. Let's just say this number here is 10. I don't know what that means, but the amount of CO2 produced by cellular respiration is 10. Then at this compensation point, you should be able to tell me what is the number of the gross photosynthesis and also the net photosynthesis. So gross is like total photosynthesis, and net is photosynthesis minus whatever's happening with cellular respiration. So at that compensation point, you should know that if we're at 0, then the amount of gross photosynthesis happening has to exactly equal what's happening with cellular respiration for it to end up at 0. So the gross photosynthesis would be 10, and the net photosynthesis would be 0. That's that number there. And then let's say we looked at this point, and this point was 5. Again, what would be the gross and net at that point? So the gross photosynthesis would have to be 15, and the net would be 5. So the net is whatever's outputting minus whatever cellular respiration is doing, and the gross is like the total amount if we were ignoring cellular respiration. Okay, question. Rachel.
Yeah, I was just wondering specifically about I think whether part 4 on that diagram is more chlorophyll limited or if it's enzyme limited.
You're going to want to think this whole area as being Calvin cycle stuff limited, and this slopey area as being light reaction stuff limited.
Okay, thank you.
Oh, you're welcome. Yeah.
On the question for number 4, I think the part where I was confused was it says that mutant has half the number of Calvin cycle enzymes compared to the wild type. However, the answer key states that the slope was also impacted, despite it not necessarily having to involve the light reaction things.
So it says half the amount?
Yes, it says half the amount of Calvin cycle enzymes, and not only did the asymptote get lower, but so did the slope for the answer key.
Yes. So yeah, so the question for people who don't know, it says, how would this graph change if you halved the number of Calvin cycle enzymes? And the unit packet answer key says not only would it plateau at a lower point, so like the total CO2 uptake at max would be lower because you have half as many Calvin cycle enzyme, half as much Calvin cycle happening. But it said you would also decrease the slope, and you would make the compensation point move to the right. That can be debated, and I would love to talk about that in detail at another time. But I did, because in class she made a big deal about how like the plateau is about Calvin cycle stuff, the slope was about light reaction stuff. So actually this morning I was looking at this unit packet question. I literally called her on the cell phone, called Dr. Roosing, and I was like, Dr. Roosing, this is what we talked about in class. This is what the unit packet said. What do you want me to say at the review? And what she said was, review packet is wrong. She said that answer is not the answer she's gonna want. If you get a question like this, where it's something with the Calvin cycle, either CO2 levels are changed, Rubisco is messed with, Calvin cycle enzymes are changed. The only thing that's going to change is the plateau point. So the correct answer is not what the unit packet says. It would want you to draw, she would want you to draw this. Everything the same, and then it plateaus lower.
Thank you.
You're welcome. So yeah, not what the unit packet says. Okay, next question.
I had two questions. So with the Calvin cycle and then so is the blue line or like blue portion, I guess, after the compensation point, that's mostly like the electron transport chain. Yeah. And so does the electron transport chain and Calvin cycle happen simultaneously? Or is it like everything occurs in the electron transport chain, Calvin cycle occurs, and then like ATP synthase?
Yeah, they all happen together at the same time, with the one caveat being like the Calvin cycle the first time couldn't happen until the first reactions. But
So then how do you like define, I guess, like these lines necessarily? Like, because these are like different portions, but if it's happening simultaneously, then like how would you know? Like, I don't know if that makes sense, but like how would you know where to like draw the lines if like everything is connected?
yeah, and that's where I was saying this is a larger debate that I could have, where I feel like there is more to be said about this. But for the fact that test is tomorrow and what she wants you to know, you can just separate them in your head, and that's going to be good enough for the test. Even though I agree that Calvin cycle is going to change that front part too. For the test, we're going to say no. Light reactions do the slope. Calvin cycle does the plateau, and that's it.
So for the problem we just did, why does the amount of, or not, I guess not amount, but like the, like, yeah, I guess like we'll just say for the amount of like electron transport change or electron transport chain change, and like why is the Calvin cycle like line still, it technically starts and ends at the same point, but it's mostly the electron transport chain, like I guess you could say like time, or it's not over time, but like the CO2 uptake becomes like lower.
Because what we're basically saying, let me redraw it because it became so ugly. What we're basically saying is from this point here, this change in CO2 uptake has to do with the light reactions. And then this point has to do with how well the Calvin cycle is working.
Yeah. So then if we bring that like line down and the light reaction, like I guess the amount that decreases, then what would that have to do with the Calvin cycle not working? Because like the Calvin cycle, technically the start and end point is like all the same. Or like how does that define as less Calvin cycle? Wouldn't that be less of the light reaction that's occurring?
If there was less light reactions, what we would expect is as we increase the light intensity, the jump in the CO2 uptake would be less, right? If the light reactions were going wrong, then as we increase light, the uptake wouldn't be as much. And if we increase the light reactions, then we would see a lot of uptake with increased light intensity. So the light reactions are changing the slope.
Mm-hmm.
and then the final amount when we're like light reactions are working as hard as they can, every chlorophyll molecule is filled, which is what that point is. Then the only thing determining how high this is now how well the Calvin cycle is working.
But then doesn't CO2 uptake usually occur in the Calvin cycle? Not the light reactions?
Yes, yes. So throughout this region, it's not that the Calvin cycle isn't happening. We're just saying that the changes to the curve in this region are not dependent on the Calvin cycle, which is it's a controversial thing to say. I'm just saying that's what we're saying for the test.
But then if we are changing that portion, then wouldn't it mean that the Calvin cycle does affect the light reaction? Because like, isn't that the part that we're changing?
If we're changing the slope.
If we're changing the slope, that means the light reaction is dependent on like how well the enzymes of the Calvin cycle are working, right?
I'm not quite sure. You're saying if we change that region, then we are changing the Calvin cycle?
Or like kind of, because if we're changing the slope, like how is changing the slope letting us know that like there's less Calvin cycle enzymes that are occurring?
It's not. It was just a problem where it said, how would the graph change if there were less Calvin cycle enzymes?
Yeah, so if it's if there were more, then how would we know to increase the slope itself, not something to do with like the plateau?
Okay. So you would change the slope if I said there was something that changed the light reaction. So if I said there was more chlorophyll.
Yeah, that makes sense. So if we're talking about less Calvin cycle enzymes, that wouldn't have anything to do with the light reaction though. So then why would we be changing the slope? Why would we
You're
be
not.
Oh.
Yeah.
Wait.
So the slope should not be changed.
Wait, but isn't the red line the slope is changing?
No, that would just be me drawing it badly. Like the slope should be the same, but it should.
Oh, it's the plateau point that decreased.
Yeah.
Oh, okay, I see. Thank you. Sorry.
No, it's okay. Yeah. So I would draw it like that, and if on the test we're like, oh, I don't know if I did it, I would maybe put like slope same, make a note to your grader. I meant for the slope to be the same. Decreased plateau. That way they know. always be clear so the grader can give you the points. Okay, yes. Next question.
I just want to confirm something with like that gross and net rate question. So could you just explain it one more times in terms of okay, this is what you have to subtract from what? Because I think can you just explain what the difference between that x-axis is and then how we know it's net and gross one more time?
Yeah. Let's draw it again.
Maybe with almost an equation for net and gross, so I can kind of visualize it a little bit better.
Yes, yeah. So we're gonna say this is 10 right here. And so in my head, so that's saying the amount of CO2 produced by cellular respiration equals 10. And the compensation point, I know that's when the stuff done by photosynthesis exactly equals the stuff done by cellular respiration. And that's why we're at 0, right? So that tells me at this point, I know that CO2 consumed or CO2 uptake by photosynthesis is 10. And that's why in total it's 0. So from that, we could get the equation of the magnitude of CO2 uptake by photosynthesis minus the magnitude of CO2 produced by cellular respiration equals the net photosynthesis. And so this would be gross photosynthesis. And so then you can imagine another point here, let's say this point is 5. So that tells me again the amount of CO2 at this point. The amount of CO2 produced by cellular respiration is 10. It's going to stay constant. Sorry. And the amount done by photosynthesis has to be 5 plus the amount it's compensating for cellular respiration for a total of 15. So we do that 15 minus 10 equals 5, which is the net photosynthesis. And if we did a point here, again, let's say this point was 10. Again, we know cellular respiration is producing 10. We know the photosynthesis gross has to be not only what the cellular respiration is doing, but 10 more than that. Well, so the gross photosynthesis has to be 20. And it would be 20 minus 10 from cellular respiration equals 10 is the net. Hopefully that helped, maybe.
Yeah, that did. So where we are talking about gross, so the uptake by photosynthesis starts at that very bottom, at that very first point, point number 1, which is the amount of CO2 produced by cellular respiration. So it starts right all the way down there for the gross.
Sorry. No. Just because at this point the light intensity is 0. So there's no photosynthesis happening. But as soon as we get a little bit of light, then
Okay.
And then photosynthesis is happening all throughout here, but it's actually losing. So we have a little bit of photosynthesis here, but it's not completely compensating for the cellular respiration. It keeps increasing till boom, here now it's exactly the amount of cellular respiration. And that's why it's cancel it out and you get 0. And then up here it starts to win. Now more photosynthesis. Photosynthesis is kind of winning against cellular respiration throughout here.
Okay, so why is it 20 then?
Why is the gross photosynthesis 20?
Yeah. So like where did you find that? Where did you conclude that value of 20?
So I know from this number that cellular respiration is 10.
Yes.
And that's happening everywhere, at every point. I knew at this point the CO2 uptake, meaning the amount of CO2 going down was 10. So I know in my head that number doesn't represent the total amount of photosynthesis. That number is the amount of photosynthesis minus what's happening with cellular respiration. So this is actually kind of telling you your net photosynthesis. And if I want to know the total, it's going to be whatever this is plus the amount it's compensating for from cellular
Okay.
respiration.
Okay, that makes sense. Okay, thank you.
You're welcome. Another question.
I was wondering
the-
about. Oh, sorry.
Go ahead.
Okay. So in photosynthesis, the CO2 that's used, is that from the environment or cellular respiration or either?
The CO2 that's used, yeah, it both comes from CO2 coming from cellular respiration and also from outside.
Okay, cool. Thank you.
You're welcome.
Would the net photosynthesis be negative in between 1 and 2 because plant is producing CO2?
Can you say that again?
Would the net photosynthesis be negative in between 1 and 2? Or the
Yes.
0?
Net photosynthesis. Yes, it would be. It would be negative. Yes, it would be negative.
Okay. And could you go over
Here.
how this is related to carbon fixation and O2 uptake?
So CO2 uptake or CO2 being consumed is accomplished in photosynthesis during carbon fixation, during the Calvin cycle, when Rubisco is actually taking CO2 out of the atmosphere. So that's why this whole unit packet question it's causing controversy, because it's the Calvin cycle is happening at every point along this graph. And so I can see the argument of why the slope and the compensation point would change. But now
for this exam, we would just say CO2 uptake is like just directly proportional, or is the only effect that affects the carbon fixation rate?
For the exam, you're going to say Calvin cycle stuff only affects the plateau.
Right.
White reaction stuff only affects the slope.
Okay.
And then compensation point and also this like y-intercept point, you're going to think cellular respiration stuff. So if you increase cellular respiration, you're going to put a point down here. And you're also going to put a point over here. If you increase cellular respiration, it's going to take more photosynthesis to compensate to actually match that range. So you should see something like that.
But that wouldn't change the slope, right?
But it would not change the slope, because the only thing that changed the slope is the light reactions and stuff with the light.
Thank you.
And again, if you have to draw, if you mean to like not change the slope, maybe make a note of that, right? Didn't change slope. All right, next question. Kara?
Oh, sorry, I don't have a question. I forgot to lower my hand. Next person can go.
Rachel?
Hi, yeah, I was just wondering if we'll have enough time to go over cyclic versus non-cyclic and versus and like when each one happens, because I'm still a little bit lost on that.
Yes, we can totally go over that. Angelina and Rainy, do y'all have questions about the graph?
I have question about this topic that we're going over.
Okay.
So, you mentioned that the chlorophyll changes the slope. I'm wondering, do we have to know anything like the difference between chlorophyll A and B?
No.
No. So they are just like chlorophyll and we just group them together.
Yes.
And then also, so all the chlorophyll located in the light harvest complex, right, in the photosystem.
Yes.
Okay. And then one more thing is about the plateau, because in lecture, I remember professor said plateau is determined by the total Rubisco present and then the carbon dioxide concentration. I'm wondering if the concentration of RUBP also changes the plateau.
Yeah, so I'm gonna say like anything in this region where it's gonna group together as Calvin cycle things. Anything Calvin cycle, so RuBP, Rubisco, enzymes, CO2 amount, all it's gonna affect where we are plateauing.
Even ATP and NADPH too.
Yeah. Yes, which is why I like putting them into these two categories is.
I see. Thank you so much.
You're welcome. Angelina.
Can you hear me?
Mm-hmm.
I had a question about when it begins to plateau. Is there any variables that change where the asymptote is on the where on the x-axis if it like shifts left or right or yeah.
When it starts to plateau, we're gonna put that in the Calvin cycle stuff category. So
got it.
Cool.
Thank you.
You're welcome.
In the lecture, I think the professor said increasing the number of chloroplasts in the plant cell will lead to higher carbon fixation rate, but increasing the photosystems in the same chloroplast wouldn't really change anything. Like, can you explain why that is?
She said increasing chloroplast would do what?
So increasing chloroplast will carbon fixation rate. I mean, that makes sense to me, but she said increasing the photosystems in the same chloroplast doesn't really change anything.
Well, increasing photosystems would increase the light reactions and change the slope. And maybe she was saying increasing the photosystems wouldn't affect the Calvin cycle.
Oh, okay, I see.
All right. All right, let's take a vote if we want to go to plant responses or if we want to talk about I guess the mechanisms of the light reactions. So put it in the chat.
Wait, do you want us to put it in the Q&A?
Yeah, because I don't think there's a comment or a chat that I can see. Okay, I'm gonna just do light reactions fast. So fast. See how fast I can do it. Okay. Let's imagine we're in a mesophyll cell and there's a chloroplast. And inside the chloroplast, there's little stacks of pancakes. And I'm gonna zoom in on a pancake. That pancake is a thylakoid. So you have the inside of the thylakoid, the lumen, and then on the thylakoid membrane, you have a bunch of proteins. You have some big protein complexes called photosystems. That's what I'm drawing here. And you have some other protein complexes. You have enzymes. And the whole point of the light reactions that occur here is we want to make ATP, and we want to make an ADPH. Why? Because these two molecules power the Calvin cycle. So that's the whole point of the light reactions. All right, so let's look at these photosystems. This is photosystem 2. This is photosystem 1. And the photosystems have different parts. Kind of around the outside, they have these light harvesting complexes. They kind of surround it all around. And in the center, they have the reaction center. So this right here is our reaction centers. Okay, so on the light harvesting complexes, there's a bunch of chlorophyll pigments. Chlorophyll pigments are the ones that absorb light. And in the reaction center, we have a special pair of chlorophyll called P680. Ugh, this is ugly. Oh well. And P700. We also have something called a primary electron acceptor. Primary electron acceptor. So the blue, that's the reaction center, and the green with all the dots, that's the light harvesting complex. I'm saying it a lot because, yes, it is actually important that you know the difference between those two. Okay, so the first thing I'm gonna do is just focus on how do we get ATP? How do we make ATP? So the first step is light is going to be absorbed. So light gets absorbed by those chlorophyll pigments. And when those chlorophyll pigments absorb light, their electrons become excited to a higher energy state. And that higher energy state is actually passed chlorophyll to chlorophyll. And as they pass that energy, it's actually called resonance energy transfer. And I call it that, and I make that distinction because they're passing energy, but importantly, they're not passing electrons. So these are not redox reactions. They're just kind of passing this weird resonance energy stuff. Eventually, that energy in the chlorophyll, in the light harvesting complex, eventually that energy gets passed to the special pair, P680. And when the special pair of chlorophyll get that energy, it doesn't just do resonance energy transfer, it does a redox reaction. So it's going to transfer an electron to the primary electron acceptor. So P680 does the first redox reaction. And redox is just any reaction where electrons get exchanged. So because it's sending an electron, that's a redox reaction. The primary electron acceptor is going to send an electron to the next protein complex called PQ, right? It's not PC. Yes, PQ. Yes, sorry, I always get PQ and PC mixed. PQ, cytochrome, PC. I think hopefully that's on your exam page, so you don't have to memorize it. So an electron gets passed from P680 to the primary electron acceptor to PQ. PQ passes an electron to cytochrome C. Cytochrome C passes an electron to PC. As those proteins are passing electrons, they don't just take the exact amount of energy they got and give that entire energy to the next person. They actually pocket some of the energy and use it. So what do they use that energy for? when they get energy from electrons, they use that energy to pump in hydrogen ions. So what we've done is we've created high hydrogen inside the lumen, inside the thylakoid, inside the pancake, and low hydrogen outside. And that's great because there is another protein over here, ATP synthase, that uses the hydrogen concentration gradient to power the synthesis of ATP. So protons are going to move down their concentration gradient from high to low through ATP synthase, and that creates a proton motive force that powers ATP synthase to turn ADP into ATP. And that's how we make ATP. So basically we get this energy from light, put hydrogen ions into the thylakoid, and use those hydrogen ions to power ATP synthase. All right, now that's all great, but we need to go back and we need to look at P680. P680 donated the first electron, and now it's not P680, it's actually P680 plus, because it's missing a negative electron. And that's bad because if we want it to keep going and keep doing this, it doesn't have an electron to donate. So we have to replenish the electrons of P680. And the way that we do that is using water. So water gets split by an enzyme in photosystem 2 into hydrogen ions and oxygen gas. And that oxygen gas leaves as waste, and animals use it to breathe. And the hydrogen ions actually help contribute to the high hydrogen concentration inside the lumen. But the most important reason why we split water is because when we split water, we can donate an electron from water to P680. And so P680 has its electron back from water, and it can keep doing these reactions. Okay, so that's how we make ATP, and that's the role of water. Now, how do we get NADPH? So NADPH has to do with photosystem 1. So it starts off the exact same. Light comes in. It's absorbed by chlorophyll molecules. They get excited. They pass that excitation in between each other.
Can I interrupt for just like 1 second?
Yes.
So doesn't water give up 2 electrons?
Probably. I don't know the exact amount. That's like chemistry knowledge.
Oh, okay. I was just wondering because if one electron goes to P680, where does the other electron go?
Yeah, no, you're probably right. I just don't know chemistry.
Okay, so it's probably not that important to know.
No, just that electrons go from water to P680.
Okay, thank you.
You're welcome. Another question?
Yeah,
So
going
is
into
is photosystem 1 necessarily not a part of non-cyclic ETC? Or are they both involved in non-cyclic and cyclic ETC?
Everything I'm talking about now is linear electron flow. Did I answer the question?
So photosystem 1 is also involved in non-cyclic.
Yeah, linear or non-cyclic involves both photosystems. And then cyclic only involves photosystem 1. Okay, so how do we get NADPH? Or do you have a question? Yes, no?
yeah, my question is just how much do we need to learn about the like cyclic part? Is it enough for us to learn that in that part there's no water broken, it's just moving of electrons around? by increasing the energy in photosystem 1, and they use that energy to pump, like, it starts around, and then the energy goes down and sent back to photosystem 1 and get more energy. Is that enough for the cyclic part?
Yes, yeah, you need to know about cyclic. I'll go over that too, but it sounds like you got it.
For where we're at right now, do we need to know about ferredoxin and like if the electrons go into that, or does that come after photosystem 1?
Yeah, I'm gonna get to ferredoxin
Oh,
right.
okay, got it. Wait, just in general, that comes after photosystem 1, or do the electrons go to ferredoxin first?
After.
Okay, got it. Thank you.
You're welcome. Yeah, so light comes in, hits the chlorophyll, they get excited. Eventually, that excitation goes to P700. Then P700, the special pair, they don't just get their electrons excited, but they actually send an electron to the primary electron acceptor. Primary electron acceptor sends electrons to ferredoxin. And then ferredoxin is going to send electrons to NADP plus reductase. And NADP plus reductase is the enzyme that catalyzes the reaction to turn NADP plus to NADPH. And now we have successfully made both of our goals. Yes, question.
So just to clarify, photosystem 2 only sets up the proton gradient so that ATP can be synthesized through photosystem 1 electron transport chain, right? Like, are they separated? Is this like, are they electron transport chain involving photosystem 2 and photosystem 1, like, are they interconnected, or is it just
Great question.
thing?
Great question. And you just kind of took me right to the last bit of my little speech, which is, yay, great, we made both of our products here, ATP and NADPH. We also made oxygen, but that's just waste. But you should be asking yourself, or the problem you should have with what I said was, okay, well, what about P700? Didn't P700 donate its electron and become P700 plus? And isn't that a problem? And don't we need water to like give electrons or something? No, it's not a problem because it's all connected. PC over here doesn't just take electrons and that's the end and they're not connected. PC gives electrons to P700. And that's why we don't need water over there. And that's why technically what starts way over here does connect all together. Other questions?
I have one question real quick. So like the electron is passed on from PS2 to PQ and then CYT. Does that mean that it's electron transfer instead of like energy transfer?
Yes, so as soon as we get past P680, we're passing electrons.
Okay.
With photosystem 1, the chlorophyll on the outside, they're not passing electrons, but P700 is, PC is, and everything downstream of P700 is.
Okay, so it's just the chlorophyll within PS2 that's energy transfer, and then the rest is electron transfer.
Yes, just the chlorophyll in the light harvesting complexes. That's the only time it's not electron transfer.
Oh, I see. So for photosystem 1 and 2, just in the light harvesting complex, that's the only place is energy transfer.
Yeah.
Okay. Okay, thank you.
You're welcome. Next question.
Is it okay if I go? So my question is regarding like the cyclic part. So I know that when NADP plus levels decrease, cyclic will dominate. And is that because there's nowhere for ferredoxin to donate the electron to, so it'll go back into PC? Or was it PQ? I just don't remember like the full cycle for cyclic.
Yeah, so let's talk about cyclic. So cyclic electron transfer is when we want to make ATP but not NADPH. Why would we want to do that? So for the Calvin cycle, you need 18 ATPs. I think it's like 12 NADPH. It could be more. I don't know if that number is right, but I do know you need more ATP. So sometimes you want to end up making ATP, but not more NADPH to get the right balance that you need for the Calvin cycle. Also, if your NADP plus levels have gotten too low, then again, you want to turn off the making of NADPH. So that's what cyclic is for. It's when you want to just make ATP. And so that is accomplished by basically only using this section. So you cut out NADP plus reductase, and you cut out photosystem too. And so it's called cyclic because what happens is light comes in, it excites chlorophyll, they excite each other, and then eventually they send it to P700. I'll do it in yellow. And P700 gets resonance energy. No, I clicked yellow. What is happening? Yellow, right? Yellow. Oh my God. Whatever. Let's do blue. Okay, so light comes in, blah, 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 goes to P700, goes to the primary electron acceptor, goes to ferredoxin. Then electrons are sent from ferredoxin to PQ, to cytochrome C, to PC, back to P700. So the cycle is P700, primary electron acceptor, ferredoxin, PQ, cytochrome C, PC, and back. And the electrons get passed in a circle constantly. Okay, how is that making ATP? Well, as you're passing electrons to these guys, what do they do with some of that energy? They use it to pump in hydrogen ions, and then those hydrogen ions get used to power ATP synthase. Okay, question? This diagram's ugly, but sorry.
So I guess ferredoxin can just send electrons either way, or just both?
Yeah, ferredoxin could send it to NADP plus reductase or to PQ. And the mechanisms of like how we decide to now all of a sudden switch, I have no idea the chemistry of that.
Wait, could you repeat one more time how it makes ATP, or like how it helps ATP synthase?
Yeah, because basically we start at PS1 and everything's the same. We end up sending an electron to ferredoxin. But then instead of ferredoxin sending electrons to NADP plus reductase, it sends it way over here, sorry, to PQ, which then sends it to cytochrome C, which then sends it to PC. And these proteins here are the ones that use that electron energy to pump in hydrogen. And when they pump in hydrogen, that creates the gradient that powers ATP synthase.
Okay, got it. Thank you. And then when you say like it helps move hydrogen in, is hydrogen just going from outside like
this cell to inside, like it doesn't need anything, right? It's just the gradient that allows it to come in.
Yeah, the gradient creates a proton motive force.
Okay, got it. Thank you.
Do you have to think about Le Chatelier's principle of redoxin sending electron to NADP plus reductase versus to PQ, or do we not have to worry about it?
No, don't say that.
Okay. Like,
Yeah, kind of fun fact is the first forms of photosynthesis only did cyclic. So it was actually like an evolution. It was first cyclic and then linear came next. That's a fun fact. All right, other questions?
Could you speak about the feedback loop of this? Like, for example, if there's like too much NADPH going on, or if there's too much ATP, like how does that affect the rest of the system?
Yeah, so the only thing you need to know about that is if you have too little NADP plus, that's going to cause cyclic to turn on. And then the other thing you need to know is the light reactions that produce ATP and NADPH for the Calvin cycle. They also rely on the Calvin cycle using those things up and then producing ADP and NADP plus. So what this means is if things start to go wrong with the Calvin cycle, that's going to eventually lead to a decrease in the light reactions. Because if we stop using up ATP and NADPH here because something's going wrong, then we'll stop making these. And if these aren't available, the light reactions can't happen.
Thank you.
You're welcome. All right, any other questions about the light reactions? It looks so crazy. All right, then let's go on to plant responses to herbivory. Okay. So for plant responses to herbivory, there's kind of two things that you need to know. You need to know 3 signaling pathways that cause chomp, chomp to activate plant defense. So there's 3 pathways we talked about that do that. And then the other big thing you need to know is how do we get signaling at different distances? So what we're going to see is the main idea of signaling, or the main player of herbivory defense signaling is JA. And so basically what this is saying is if we have a bunch of plant cells and chomp chomp, caterpillar ate right here, how do we get JA signaling locally? How do we get it to nearby cells? And then how do we get JA signaling to happen to really far away cells? If this is where the damage happened, how can we get signaling at all these different distances? That's what you need to know for the test. So what are the 3 different signaling pathways? They are HAMPs, DAMPs, and HIPVs. So all of these happen. We have little caterpillar that just chomp, chomp. When the caterpillar eats a part of the plant, all of these things get activated. So what are they? HAMPs are herbivore associated molecular patterns. DAMPs are damage associated molecular patterns. And HIPVs are herbivore induced plant volatiles. Those are kind of our three different signaling molecules that come up in response to this guy. So HAMPs, you can think of like herbivore secretions. So when they actually eat the plant, they're going to produce some kind of secretion. And an example would be saliva. So there are molecules in the saliva that are HAMPs. DAMPs, you can think of cell damaged spewing guts. So when they chomp down on the plant cell and the cell breaks and it spews all of its contents, some of that stuff that gets spewed out, those are DAMPs. And an example is amino acids. There's just like a bunch of amino acids inside of cells. And if you break open the cell, some of those amino acids are going to come out. And then HIPVs, an example is, well, they're all going to be gas signals. And that's all I'm going to say about HIPVs for now, and we'll come back to them. But let's focus on the signaling pathways for HAMPs and DAMPs. So let's say the plant cell gets damaged. There's some saliva. What that saliva is going to do, let's say, I don't know, something in the saliva. Maybe it's like enzymes. I don't know. Saliva, digestion enzymes, whatever. Some molecule in the saliva is going to activate elicitor receptors on the plant cell. Those receptors on the plant cell are going to activate a signaling cascade, aka we don't know the details of what happens, but we do know at the end we get an increase in jasmonate or jasmonic acid or JA, and that's kind of the key plant defense signal. Jasmonic acid is going to enter into the nucleus. And in the nucleus, the JA is going to bind to its receptor. Then jasmonic acid plus its receptor, JA plus receptor, binds and kicks off JAZ. So what is JAZ? Well, if you looked at DNA, let's say these are my plant defense genes. JAZ is a repressor, repressing the transcription of those genes. JA plus its receptor kicks off that repressor, and the end result is to, oops, I didn't even do that, increase plant defense gene transcription. So we had these salivary molecules, end result, increase plant defense gene transcription. And that's a lot of different things, a lot of different responses in the plant that will cause it some kind of defense. But if you're like, can you make that more concrete? An example of what one of these plant defense genes would do is to make a protease inhibitor. Well, what does that do? Proteases are really important to herbivore digestion. Proteases are how herbivores digest proteins. And if you inhibit their proteases, they can't digest proteins. So this is really bad for the herbivore. And for bugs, it's actually, it's going to kill them. If they can't digest proteins, they're going to die. So this is a pretty good defense for the plant. But there's lots of other things that happen. So another one that you should know is that it's going to, oops, I didn't mean to put S. It's going to create enzymes that synthesize secondary metabolites. So how's that a good defense? Well, secondary metabolites are like, they're basically just, they contrast primary metabolites in that they're not essential for the plant cell. So there are things the plant cell can make, but it's not essential for the growth and the life of the plant cell. But specific secondary metabolites that are important for defense would be like, there are some toxins that they make. And also, importantly, you should know HIPVs. So those gas signals that we're also going to talk about as one of the 3 main signals, that comes out of plant defense gene transcription increasing. Yeah, question.
Would we classify tannins as a toxin as an enzyme?
Yeah, tannins are a toxin that's produced as a response.
Okay, thank you.
You're welcome. All right. So that's one signaling pathway that you should know. What about the DAMPs? So this is responses not to the saliva of herbivores or some kind of secretion that they do, but to a cell actually breaking open. And the example would be amino acids. So it's actually going to be the exact same. It's going to be the amino acids are going to activate an elicitor receptor, cause a signaling cascade that increases JA, that in the end, blah, 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 kicks off Jazz and increases plant defense genes and does the exact same stuff. It's the exact same thing. The only difference is now we know the details of the signaling cascade. So you can thank those scientists because now you have to know it too. So you can't just say signal cascade, you have to know the pathway. So what is the pathway? Amino acids are going to activate the elicitor receptor called the glutamate-like receptor. And the glutamate-like receptor is on the plant membrane, and it's a channel that lets through calcium. And so that's going to cause an increase in calcium influx into the cell. That's going to do two different things. It's going to, number one, it's going to depolarize the cell. And it's also going to activate calcium gated chloride channels. So these calcium gated chloride channels are actually going to let chloride leave, which you should maybe kind of think in your head, okay, this is different than what happens in plant growth. In plant growth, we want chloride to go in to make more solute in the cell. What's happening here is chloride leaves, so we actually get more depolarization. And all this depolarization is then going to also activate voltage gated calcium channels. And those voltage gated calcium channels are going to increase calcium. So in the end, we get all of these things happening that activate a bunch of channels that cause a ton of depolarization and a ton of calcium. Before I move on, is there a question?
I had a question about HAMS and DAMPs. So would we consider HAMS as only local and DAMPs as systemic? But then do HAMPs cause DAMPs? And is that why there's signaling cascade also written down in the HAMP section?
No. So definitely I would not consider either one of these only local or only systemic. And I'll get to, how they signal at different distances, how all of these relate to those different distances too.
Okay, thank you.
You're welcome. So right here, all I'm saying now is like, you need to know the specifics of this signaling cascade and all these receptors and all the channels they activate. And in the end, this increase in calcium, what does it do? It increases jasmonic acid. And then the exact same thing happens. Jasmonic acid goes to the nucleus, it binds its receptor, it kicks off JAS and increases defense genes. And that it makes protease inhibitors, or it makes toxins, or it makes the enzymes to make secondary metabolites like HPVs. All of those things happen also to DAMPs. It's just now we know this specific stuff. Okay, now before I move on, I'm going to start to talk about how does this all have to do with distances, right? So you can imagine, I don't know, say we have some plant cells here. Everything I've said so far about the HAMPs and DAMPs, it's like extremely local distance. So if the damage happens right here, that's where the saliva is. That's where there are broken cells. So right there, we're getting these responses, these increases in JA right nearby. But we can also get a spread of the signal to cells nearby and cells even further away. So how is that accomplished? So first, let's think about how do we spread the signal to cells nearby? And the answer to that is going to be through electrical coupling. So cells nearby can also increase their JA signaling through electrical coupling. So that all has to do with this mechanism we just saw here from the DAMPs. So we get this damage and our plant cells can actually electrically couple because they're connected by plasmodesmata. So all that damp signal originally happens at the damage site. If we get this first cell depolarizing, that depolarization signal can be sent through plasmodesmata to nearby cells. And those cells have their own voltage gated and calcium gated channels. So they can actually re-up that depolarization. And they can send that signal to the next cell. And that cell has all those channels and can also do that depolarization, can also increase calcium. And because they can all do that, even though the damage happened here, we can get JA signaling through that electrical signaling happening in cells not directly at the damage site. So that's one way we can get signaling nearby. Yeah, question.
I'm still a little bit confused on why we separate amino acid versus HAMPs when the signaling cascade is still there for both. So then what exactly is the difference?
Yeah, they're both happening simultaneously. We're making a distinction because HAMPs are activated by molecules in herbivore secretions. So the herbivores secrete stuff when they're eating. The most kind of obvious one is saliva. And things in the saliva activate the HAMP pathway.
Oh, okay, thank you so much.
Yeah.
That makes sense.
HAMPs are activated by cell damage stuff. Yeah, other question.
So it's safe to say that HAMPs do not have the GRLs.
The what?
The GLR protein, so they don't have that?
Yeah, so we're saying that we know amino acids do this specific pathway, and we don't know the details of the HAMP pathway.
Okay.
So it's possible maybe GL are involved, but we don't know. Scientists haven't figured it out yet.
But like if there was a question, right, and we were asked to be given the like pathway, and it specifically like we could identify if it was a HAMP or DAMP, we would then give if it was like a HAMP. So if it obviously the question said like saliva, we would just say signaling cascade then to JA. But if it was like a damping, like a well, because like if the question was like, oh, animal like ate a plant, right? Then that's both. So
Yeah.
then what would it say like we could do? I mean, that I'm getting like really specific, but would we know what pathway to draw?
Yes, we would narrow it down for.
Okay.
Yeah.
Okay. All right. That makes sense. Thank you.
You're welcome. Okay, the other way we could get kind of nearby cells to have JA signaling, even if they're not right at the site, is also through those plasmodesmata, the JA molecules could move through there too. So I don't think that would move very far. It would probably be really local. The electrical signaling is going to allow cells kind of further away to get the signal. But just keep in mind that JA can also move through the plasmodesmata to nearby cells. But if you're asked, like, how can we get signaling in a nearby cell? You should be like, this electrical signaling is how we're going to do it. That's how we're going to get it to those cells way over there. Okay, so we can get really local signaling right at the cut site. We can get a little bit farther out using electrical signaling, but how do we get super far out? That's where the HI PVs come in. Those are going to be for causing JA signaling in cells super far away. So where do the HIP V signals come from? Those are going to be the results of plant defense genes. Being transcribed. Ah. So at the actual damage site, you would have to have these two things happen first that then result in the enzymes for making secondary metabolites be made in order to get those local cells to make HIP-Vs. And then those local cells are going to secrete these HIP-Vs as gases. That's what volatiles are. Means like they're very prone to becoming gases. So locally, those local plant cells are going to produce the HIP-Vs, and then those gas signals are going to move super far away. And in a plant cell super far away, the HIP-Vs are going to bind the elicitor receptor. And guess what? Yay, we don't know the pathway. So we're just going to say signal cascade. Increase JA. And then all the same JA stuff you should be able to say too. But the important thing is this, what makes this different is that it's happening in a cell so super far away from the damage site. So those are the three signaling pathways, and the different distances you should be able to explain is like at the actual cut site, at nearby cells, and then at cells super far away, maybe even on a different plant. Okay, Maya, yeah.
Yeah, so JA can also travel through phloem, right? And bulk flow through the plant. So is that considered nearby or also like farther away?
Yes, that would be considered far away.
Okay.
So yeah, not only can JA go through plasmodesmata near, but it can also travel through phloem. So thank you, because I was going to forget to mention that. So in addition to HIP-Vs, JA going through phloem, traveling through those tubes would be a way for JA signaling to reach super far away. It's like the one time xylem and phloem comes up.
Wait, I just had a quick question, just a clarification. For that, like someone asked the question, I think, like if they say an animal bit, like a leaf or something, how do we know it's a hamper damp again?
If it just said animal bit here, explain what happens, you could choose either one.
Okay.
We would narrow it down for you and say, if it was a saliva secretion, then you would say it's a hamp. But you could say hamp/damps, you could say, or we would narrow it down, and if it was a cell damage, you would say damps. If it was like herbivore saliva, you would say hamps.
But then if we write hamp/damp, won't the signal cascade be different? So then how do we know what to write beyond that?
I mean, if you write hamp/damp, yeah, you would.
So we would technically have to choose one. Like if they make us write like a flow diagram, we couldn't necessarily write hamp/damp, right? Because I'm guessing for the test they'd want something that's in detail, so we would have to write about like the GLR and all that, and we wouldn't be able to just write signaling cascade.
Yeah, I'll say that on the test, it narrows it to where you'll know which one.
Okay, sounds good. Thank you.
And so, yeah, you should know both, and you should definitely know how to say everything that happens with JA to the point of plant defense gene transcription.
Okay, and then sorry, could you repeat one more time what the function of a protease inhibitor is?
Yeah. So a protease is an enzyme inside the bugs, inside the herbivores, that are really important to allow them to break down proteins. So it's really important to their digestion. If they don't have proteases, they can't digest proteins. So the plants are like, I'm going to make a protease inhibitor. And you can say goodbye to your proteases because they're not going to work anymore.
Okay, perfect. Thank you.
You're welcome. Yeah, other questions?
I was just wondering specifically if it's possible to start like only a HAMP signaling pathway. Let's say if the herbivore just drooled on the plant, would the chemical be in the way, or is it possible to induce an only HAMP response?
No, yeah, yes. That exactly does happen. Sometimes the bugs are doing things on the plant that are actually not damaging it, but they'll induce these responses that way. So yes, yeah.
All right, thank you.
Yeah, Angelina.
I wanted to ask a question about the HIPVs. So you say that after HAMP and DAMP, the plant defense genes are transcribed. What do you mean by bind elicitor receptor?
I'm just saying that these molecules, HAMPs, DAMPs, HIPVs, they have receptors in plant cells. And if we don't really know what they are, we just call them elicitor receptors.
Got it. And then you say that it's the same as HAMP and DAMPs for like the JA goes into the nucleus, binds to the receptor, but in HIPVs, then does it create more plant defense gene transcription in the plant that it has now signaled?
Yeah.
Okay. Got it. Thank you. So it's kind of just like stuff in the air, the gas, and then it triggers.
Same thing.
Yeah, got it. Thank you.
You're welcome. Another question? No? Cool, yeah. So you should know these pathways and know how we do different distances. Any other questions about plant herbivory? All right, last topic of the test. We'll go over it super quickly, is thyroxine. So this is like the one non-plant thing we did. We went over basically a just illustrative example of hormone signaling in animals. And we picked thyroxine. And thyroxine is a hormone that when it's secreted by the thyroid, it signals to all the cells of your body to increase metabolism and other stuff. But what you need to know for the test is how thyroxine is synthesized, how it's regulated, and what happens when the regulation goes wrong and you get too much thyroxine or too little thyroxine. So first, let's talk about, you should know like the steps of how thyroxine is synthesized. So thyroxine is synthesized in the thyroid, and you need two things to synthesize it. You need thyroglobulin, and you need iodide. So those two things you need. So in the thyroid, you have these little follicle cells here, and you have the inside lumen, and both are going to be involved in the synthesis. So in the follicle cell, we make thyroid globulin, which is right here, and then we send it out into the lumen. So we're going to end up kind of putting together thyroglobulin and iodide in the lumen. So that's how we got thyroglobulin out there. Now, how do we get iodide over there? So iodide is going to come from the blood. It's going to be over here in the blood. And then we're going to send it into the follicle cell using a symporter. So we use the movement of sodium, high to low, to move iodide against its concentration gradient from low to high into the follicle cell. And then it moves straight through the follicle cell and goes through a channel to get to the lumen. It's called a Pendron channel, and that's moving just facilitated diffusion high to low. Okay, once we have both thyroglobulin and iodide in the lumen, what happens next is we're going to turn iodide into iodine. We're going to oxidize iodide. And then once it's oxidized, we can combine those two things together. So iodine plus thyroglobulin get combined together. And the big molecule that we make basically has pieces of T3 and T4. And so T4 is thyroxine and T3 is the modified kind of active version of thyroxine, but they're all kind of attached together. And then they just kind of hang out in the lumen there until the thyroid gets activated. When the thyroid gets activated, it's going to want to actually release thyroxine, release T3 and T4 out into the blood so it can go and signal to the rest of the body. So when the thyroid gets activated, then we're going to try to move this big molecule with all these pieces of T3 and T4 back into the follicle cell. Once it's in a vesicle in the follicle cell, we'll break up that big molecule into individual pieces of T4 and T3, and then it will be secreted into the blood. And it'll go on and signal. So how does the thyroid get activated? There are different things that can activate the thyroid, but one big one you should know is TSH. So there are TSH receptors on the follicle cell, and if TSH comes into the blood, binds the receptor, that's going to activate the thyroid. That's going to cause this movement inward and movement of T3 and T4 into the blood. Right, question? Can I see a question? Yeah.
I just had a question about the iodine going into the lumen. Where does DuOX2 come in?
Yeah. DuOX2 is the enzyme that seems to be involved, at least in pandas, in converting iodide into iodine, or oxidizing iodide, making it uncharged.
Okay, so for some reason I thought it was like that was the channel that brings iodide into the lumen, but it's not, it's the Pendron.
Yeah.
Okay, all right, makes sense. Okay, thank you.
You're welcome. Okay, so that's how we synthesize thyroxine and release it once the thyroid gets activated. Other things you need to know. How is this regulated? You should know there's the hypothalamus question.
Sorry, I was just wondering, does TSH, does it have a direct effect on like the iodide that's coming in, or does the TSH that's only causing the T3, T4 to leave the lumen and go out into the blood?
Yeah, the TSH receptor activates the thyroid, and when it activates the thyroid, that's signaling to the thyroid, oh, I need to release T4 and T3. And in order to do that, I need to bring this guy in, I need to break it up, and then I need to release them. So the TSH doesn't really affect iodide, it affects this here.
Okay. And then does the iodide just, is there like some kind of driving force that's leading iodide to keep on coming into the thyroid cell, or is it just like always like that, like at rest?
Yes. So first we need to have enough iodide in the blood just to start off. And then what makes it keep going in is the fact that we have the correct sodium concentrations. So it's the sodium movement that is the driving force of iodide in to the follicle. So
Okay, got it. Thank you so much.
you're welcome. Question?
Yeah, I was wondering, how does T3 and T4 get out of the follicle cell again? Is it through a channel?
Here?
Yeah.
So from what I read in the textbook, is that it was exocytosis, but in this diagram, it's looking like it's through some channel. I don't know. Question mark. All right, regulation. So there's hypothalamus, interior pituitary, and then our thyroid. So the thyroid's the one that makes the thyroxine. How do we regulate that? So the hypothalamus releases TRH, which positively activates the interior pituitary. I can't speak anymore. And the interior pituitary releases TSH, which positively activates the thyroid to release T3 and T4. And then these outputs of the thyroid, they inhibit the interior pituitary, and they inhibit the hypothalamus, which is negative feedback. It basically means as the products get high in concentration, the signals that cause them to be made are going to be turned off so that we never get those levels too high. And so you can expect or imagine questionable tests where we say, what if we decrease TRH, or we decrease TSH, or we inhibit this pathway? What would the results be? So you should be able to predict changes to this pathway. And the last thing you should know is what happens when you have too much thyroxine or too little thyroxine being produced. So too little, that's called hypothyroidism. Too much thyroxine is hyperthyroidism. And there's lots of different things that can cause it. And probably on the test, you can expect one you haven't heard of, but it's going to be something that, based on changes to these pathways, results in too much thyroxine or too little thyroxine. Yeah, Rachel?
I just a quick question about, I think, goiter formation. I think this was in regards to hyperthyroidism, but I just wanted to know for the reason, sorry, I think this is hypothyroidism. I was wondering, does TSH also stimulate the growth of the thyroid and cause it to physically get bigger? Or is it because there's just more thyroglobulin being produced at a higher rate than it can be exocytosed?
Yeah, so if you stimulate the thyroid too much, just by itself, too much stimulation can cause the thyroid to grow in size. It doesn't necessarily have to be a buildup of thyroid globulin. All right. So yes, next thing is too much, too little. You should be able to predict how those things happen because you understand the pathway now. And you also need to know how both can result in goiter. And goiter is enlarged thyroid. So we'll go over how that can happen for both. So that happens with hyperthyroidism, like we just said. If you have too much thyroid stimulation. So if the thyroid is overactive and you have all the stimulation, that can lead to an enlarged thyroid. So an example of that would be the TSH receptor. Let's say it mutated to be always active. So even if there's no TSH around, it's always signaling to the thyroid, keep producing, keep making more thyroxine. All of that activity causes too much stimulation and an enlarged thyroid. Okay, you can also have too little thyroxine causing goiter. And an example of that would be iodide deficiency. And what happens there is you have a buildup of thyroid globulin. Globulin. So if you don't have iodide around, it won't attach to the thyroid globulin, but everything else will still happen. So it'll still intake the thyroid globulin, but it's not going to make functional T4 or T3 because there's no iodide. So it just builds up inside the cell and it enlarges the thyroid with that buildup. Yeah, Kira.
I had a question about just like the goiter and like the enlarged thyroid in general. So like on the practice questions, when we were doing when the difference between hyperthyroidism and hypothyroidism for the goiter, it always kind of circled back to an increase of thyroglobulin. And so does thyroglobulin always have to make that thyroid, like that goiter increase? Like, you know?
Yeah,
Is it always that? Okay.
Do you have to get a buildup of thyroid globulin?
Yeah, do you always have to have that to have a goiter?
No, so you can get hyperthyroidism, like in this case right here, if the TSH is always active, but there's still enough iodide around, then everything's going to be normal, right? We're still going to attach iodide to thyroid globulin and we're going to end up making a bunch of T4. So you're not going to necessarily get a buildup of thyroid globulin, but you can still get goiter just from too much stimulation of the thyroid.
Okay. Because I think like in the question for the like pathway specifically, it was like, okay, too much TSH stimulation, yes. But then what it explained was like, oh, too much TSH then creates increased thyroglobulin production, which then leads the like an excess of thyroglobulin to how much oxidated iodide is in the lumen. And then it was like, oh yes, increase, like, and then that still resulted in an increase of thyroglobulin. So I was like, is that always going to be the case? So and you're saying it's not.
No, but it can
Okay.
also be a cause. Yes.
Okay, that makes sense. Okay, cool. All right, thank you.
You're welcome. Okay, that's it. Any other questions? But that is everything that is covered on the test. So unless you have other questions, that is all.
So, what are the 4 things again? I know we went over a lot. I kind of lost track of what we started with.
Me too. What did we do first? Oh yeah, okay. The first one was elongation. So
Like
water potential stuff and auxin signaling, phototropin. That's 1. So I just group that as like plant elongation. 2, photosynthesis. You should understand that graph very well. You should understand the mechanisms of the light reactions and cyclic and versus linear. The 3rd was plant responses. Know those 3 different pathways and also how you get different distance of signals. And then 4 was thyroxine. It's regulation.
Awesome, thank you.
Yeah.
Sorry, I'm asking so many questions. But when
Oh.
it comes back to photosynthesis, we spent like a lot of time on the light reactions and like the specific details of that. When it comes to the Calvin cycle, do we need to know all that much detail except for like Rubisco?
Exactly. You should know Rubisco does carbon fixation and that the output is G3P.
Okay. All right. Perfect. Thank you.
You're welcome.
How much about pandas do we need to know, like in specific?
All of those like experiment details, you don't need to know. Just what in terms of like what we went over today, but no specifics.
Okay.
Anything.
And then what was, I just want to clarify, what was the thing that you said to know the difference between linear and then what was the other one?
Linear and cyclic electron flow in the light reaction.
Okay. Right. Sorry, I thought you meant for the graphs, but thank you.
You're welcome.
For the lab that,
yeah,
for the lab that was this week that some of us missed, is that material going to be on exam 4?
What was this lab? What do we do?
I think it was the cucumber seeds.
Oh, okay. So basically what they did was they measured the size of the plant growth in light versus not light, and then the board work was just the auxin signaling pathway.
Okay, cool. Because I normally look at the lab follow-up stuff, but it was just the data, so I was like, no, I had no idea what was going on. So
yeah, as long as you know the auxin signaling pathway, there's nothing from the lab that's relevant besides that.
Thanks.
You're welcome.
Hi, Rachel. I have a question. So I was doing like the practice exam question from this week's practice exam quiz, and it's asking about what color of light inhibits the whole stem elongation, and the answer is red. And the photopigment, the answer set is phytochrome. I don't remember that in our vocabs. I'm just wondering.
Yeah, I was not there on Friday, so I'm not sure if that was gone over in lecture, but I can tell you it's not going to be relevant for this test. So
okay. And then one more is the phototropism. What is that on again? I forgot. Like, is it the photosynthesis chapter? Or
phototropism is a plant responses to light. That's what that literally means. And then phototropin
I see.
is a pigment molecule that absorbs light and then has the output of moving pin proteins as a cell.
Oh, that would be the PHOT part?
Yes, that's phototropin.
Oh, I see. Okay, got it. Thank you so much.
You're welcome. Question.
So Rachel, we went over a lot. I noticed that it's recording. Would you be able to get us the recording by tonight so we can review?
Yeah, I think it takes some amount of minutes to load, but I can put it up tonight. Yes.
That would be awesome. Thank you.
You're
Thank you
welcome.
so much for doing this.
Of course. Of course. Yes, question.
So for the phototropin specifically, I remember that it only absorbs blue light. And so how is that like significant in any way, the that it only absorbs blue light? And also, like just as a hypothetical, what would happen if like there was no blue light at all and it only absorbed like every other wavelength except for blue light? Would the elongation just like not happen at all?
Yeah, so it is relevant not to this test because I mean I wasn't there on Friday, but for people who were there, y'all didn't talk about the fight pigment or did y'all talk about like long red light versus on regular red light versus shade responses? I don't think it comes up on the test. I mean, not that I don't think. I know it doesn't come up on the test. But anyways, yeah, it is a big deal and it is relevant for signaling, but not for the test. Basically, if there isn't blue light around, there's other signaling pathways that move pin proteins differently to different places. Like when there's kind of shade all around, the pin proteins get moved to the bottom. And that's why you saw the plant in the shade in lab have its shoots really extend because of the movement of those pin proteins. Sorry, not to the bottom, but to the sides. So they got their auxin biased into the shoots instead of going down to the roots. But that's, I think she's going to talk about it next. Yeah, on next exam.
Okay, cool. Slay.
Question.
So I think I might be confusing myself more and more. But what light, like what wavelength of what color of light do plants absorb again? I think red, like orange and blue, that three.
This is a good question. And I think I know where you're getting confused. So plants have many different proteins for absorbing light. The ones for photosynthesis, the chlorophylls, they like bluish, purple, and red. Now there's completely different proteins, not photosynthesis involved, that are pigments for signaling things about growth, like phototropin. And that one really likes blue and doesn't like red. And then there are other ones we're going to talk about that like red. So there are different ones for different things. But none of them like green. I hope that helped.
Wait, so for photosynthesis, only red-ish that kind of lights work, and for growth, only blue-ish lights work.
No. Remember, there was two peaks for photosynthesis.
Right?
Blue
Yeah.
So blue and red's good for photosynthesis.
Oh, but blue. Wait, but for growth, blue is the only thing that works.
From what we've learned so far, yes.
Okay, awesome.
Because y'all didn't learn about fight, right? PHYT? Did y'all?
I think Jake mentioned it just by a little bit.
Okay.
It was in the reading in the textbook, but I don't think it was in lecture that much.
Okay. Yeah, question.
I had a question about your diagram for the cell signaling pathways for plant growth and elongation. If you could scroll back up to that. It's the one with like it's like a grid almost.
Mm-hmm. Maybe. Okay, okay. Oh, was that it?
Yeah, that one. Yeah, that one. I wanted just to clarify what the red dots were. I didn't know if they were the phototropin or the pin proteins.
Pin
proteins. And then what you mean by the pin proteins move to the opposite side of the light. So are the phototropin, they're not in this diagram, right? Just to
They're
double
not,
check.
yeah.
They're not. Okay.
If I
Got it. Just wanted to double check that. Yeah.
I kind of zoomed in on it.
Oh, okay.
And I'd say they moved on this side, on this side, on this side. The light's coming on over here.
Okay, got it. Because one of the videos from the external brain was the person showed one diagram of one singular cell. But now I understand, now that you zoomed in on it. Thank you.
Of course. All right. This is your last chance. Going once. Going twice. All right, question.
Sorry. I have like one question about, so this question talks about like for treatment of cucumber seedling stems in water. And I'm just wondering, what's the difference if the kinase is blocked and treatment A, the seedling stems is put in neutral pH water, and treatment B, the seedling is put in acidic pH water?
So when I'm hearing this, I'm thinking, okay, I'm talking about growth stuff. So I'm thinking about stuff, thinking about acid stuff, and I'm thinking about pump stuff. When you said kinase phosphorylates things. I need my H pump to be phosphorylated. If I mess with the kinase, I'm going to mess with the ability of the hydrogen pump to be phosphorylated. And if you mess with that, then you're not going to pump hydrogen out into the cell wall, and all that stuff's going to mess up. Then if I add acid to the outside, then I'm basically putting protons into the cell wall. So thinking about how those things would affect, basically.
That means there will be more elongation. Like the cell will be loosened up more.
Yeah, acid into the cell wall would increase the extensibility. If you I don't know what happens to the kinase, I forgot what you said, but
the answer actually said there will be greatest elongation rate in neutral water.
Neutral water. What was the thing with the kinase?
The kinase is blocked inside cell.
So kinase is blocked, so the H pump can't be phosphorylated.
So it'll be inactive.
And it's saying it would increase growth with that? That seems, well, that should decrease growth.
Maybe I'm reading something wrong. I'm sorry.
No. Anyone else know which one she's talking about? Maybe.
I think I got it now, because actually it's talking about how phosphatase block occur inside the cell plus acidic pH that will lead to a greatest elongation.
Yeah, okay. So phosphatase, is the opposite.
So the difference, the difference between neutral and pH water, like lower pH water, lower pH water just increase like more cell wall to be loosened up.
Yeah.
Because there. Oh, okay. I see. Thank you.
You're welcome. All right. I will upload this for you guys, so keep a lookout so you can watch this again if you are, I don't know, crazy. But bye.
Thank
Thank
you.
you, Rachel.
Rachel.
Of course, of course.
Thank you.
Thank you so
Thank
much.
you.
Of course.